Welcome to video series on protein metabolism. Is on 1-carbon metabolism and 1-carbon. Now what you will learn in this video? So 1-carbon metabolism in a very simplified way, interconversion of 1-carbon compounds, donors and acceptors of 1-carbon metabolism, important MCQ discussion, probable university based exam questions and memory trick for revision. Now these are the 1-carbon compounds, and these include the formyl group represented by CHO, formimino represented as CH double bond NH, methynyl group represented as CH, hydroxymethyl group CH2OH, methylene CH2, methyl represented by CH3 group. Among these, all these compounds are carried by folic acid. Its active form, this tetrahydrofolic acid, carries all these compounds except methyl group is carried both by folic acid and vitamin B12. So this is one of the Viva question, examiner's favorite question. So methyl group is carried both by folic acid and B12, whereas all other group, that is formyl, formimino, methynyl, hydroxymethyl, methylene, methyl, are carried by folic acid. Okay, so this is how quick easy mnemonic to remember this 1-carbon compounds include FM Hyderabad. So this stands for formyl, formimino. M stands for methynyl, methylene, methyl, and Hyderabad stands for hydroxymethyl. So easy mnemonic to remember 1-carbon compound. Coming to interconversion of 1-carbon compounds, the formyl group undergoes dehydration with removal of water molecule to form methynyl group. Now this methynyl tetrahydrofolic acid can also be formed from formimino group. So this methynyl group is reduced to methylene group, and this methylene tetrahydrofolic acid is also formed from hydroxymethyl tetrahydrofolic acid. Further, methylene and tetrahydrofolic acid is reduced to methyl tetrahydrofolic acid. Okay. Now this methyl tetrahydrofolic acid donates a methyl group to active form of B12, that is cobalamin, and it becomes methyl cobalamin. So this in turn donates the methyl group to homocysteine, which gets converted to methionine. Now methionine gets converted to its active form S-adenosine methionine with the donation of adenosine group by ATP, and this S-adenosine methionine, the active form of methionine, donates a methyl group to any acceptor in the body, which becomes methylated acceptor, and it gets converted to S-adenosyl homocysteine. So this gets converted back to homocysteine, and homocysteine can be converted back to methionine with the help of methyl group being donated by cobalamin. So this is how the 1-carbon compounds are getting interconverted in our body. So the question can be asked in this format: describe the interconversion of 1-carbon compound. Straightforward 5-marker question. So easiest way to remember this, if you cannot remember this entire flow chart, at least try to memorize this part. Formyl gets converted to methynyl, methynyl is reduced to methylene, which gets further reduced to methyl group. So methynyl is also formed from formamine group, and methylene is formed from hydroxymethyl group. So first remember the skeletal part, then you can memorize this part of the flow chart. Coming to donors and acceptors of 1-carbon compound. Now this here this green ink represents the donors, and this red ink words represents the acceptors of 1-carbon compound. Now formyl tetrahydrofolate is formed during metabolism of tryptophan. Tryptophan is acted upon by pyrrolase enzyme, and this forms N-formyl kynurin, and the formyl group is donated to tetrahydrofolic acid, and it forms formyl tetrahydrofolic acid, and the acceptor is C2 of purine. So just check out here, the C2 carbon atom of purine bases adenine guanine is contributed by formyl tetrahydrofolic acid. Now this formyl group undergoes with removal of water molecule, it forms methynyl tetrahydrofolic acid, and this methynyl tetrahydrofolic acid is also formed during metabolism of histidine. Histidine is acted upon by histidase enzyme to form urocanic acid, which is acted upon by urocanase enzyme to form imidazole propionic acid. This is acted upon by hydrolase enzyme to form formiminoglutamate acid. The formimino group is donated to tetrahydrofolic acid, releasing glutamate. Now this formimino tetrahydrofolic acid can get converted to methynyl tetrahydrofolic acid, and this methynyl group forms C8 of purine bases, that is adenine and guanine. So this is check out, C8 of purine is formed from methynyl tetrahydrofolic acid. Methynyl group can be reduced to methylene tetrahydrofolic acid. Now this methylene tetrahydrofolic acid is also formed from serine and choline. So serine and choline initially forms hydroxymethyl group, which gets converted to methylene tetrahydrofolic acid. Now this methylene group is is used to form deoxy UMP to deoxy TMP. So as you all know, thymine is the special base present only in DNA, and this is formed from uridine base only. So now this deoxy UMP accepts the methylene group from tetrahydrofolic acid and get converted to deoxythymidine monophosphate, and tetrahydrofolic acid gets converted to dihydrofolic acid. It is reduced back with the help of NADPH, and methylene group is donated by serine, and serine forms glycine, and tetrahydrofolic acid gets converted to methylene tetrahydrofolic acid. This is how it gets regenerated back, and during this conversion serine can be converted to glycine also. Now, this methylene tetrahydrofolic acid further gets reduced to methyl tetrahydrofolic acid. Now, this methyl group is donated to cobalamin, which becomes methyl cobalamin, and this methyl cobalamin donates the methyl group to homocysteine, which becomes methionine. And methionine, as you all know, it's a methyl group donor. It donates methyl group to any acceptors, which becomes methylated acceptor. For example, guanidinoic acid accepts a methyl group from methionine and gets converted to creatinine. Now, this creatinine undergoes phosphorylation to form creatine phosphate, which is stored in the muscle, and it is used to utilize for muscle contraction by Lohrmann's reaction. Histidine undergoes methylation to form methyl histidine. Serine undergoes methylation to form choline. Phosphatidylcholine is lecithin, which acts as lung surfactant. Similarly, ethanolamine also undergoes methylation to form choline, which phosphatidylcholine, as you know, it is a component of biomembrane, also component of lung surfactant. Acetyl serotonin undergoes methylation to form melatonin, and this melatonin is responsible for maintaining circadian rhythm, sleep-wake cycle. Norepinephrine undergoes methylation to form epinephrine, which is responsible for fear, flight, and fight response. Carnosine gets converted to anserine. So these are the methyl group acceptors. Okay. Now the question that can be asked is describe donors and acceptors of one carbon compound. So the easiest way to remember this is formyl group. This formyl group is donated from metabolism of tryptophan, and the acceptor is C2 of purine. Formimino group comes from histidine metabolism and gets converted to methinyl group, and the acceptor is C8 of purine. So hydroxymethyl is formed from serine and choline, which gets converted to hydroxymethyl group gets converted to methylene group, and the acceptor is glycine, and also for the formation of thymidine monophosphate. And the methyl group acceptors include histidine, guanidinoic acid, serine, ethanolamine, acetyl serotonin, and norepinephrine. Okay, so these are the donors and acceptors of one carbon compound. So this was all about one carbon compound. This is a very important topic. It can be asked as 5-marker question and also long essay. Now let's see how good you have understood the topic. The main carrier of one carbon unit in metabolism is tetrahydrofolic acid. So it carries all the carbon groups like formyl, formimino, methinyl, methionine, methyl. Whereas methyl is carried by also. One carbon metal is primarily associated with nucleic acid synthesis because it is used for the formation of C2 of purine, C8 of purine, and also conversion of deoxy UMP deoxy TMP. So the active form of folic acid is involved in one carbon metabolism is tetrahydro. Okay. So the conversion of homocysteine to methionine requires what? Vitamin B12. Okay. So deficiency of B12, what happens? Homocysteine does not get converted to methionine, and there occurs homocysteinemia and homocysteine. Universal methyl group known in the body is S-adenosyl methionine, active form of methionine. One carbon units are commonly derived from all of the compounds except leucine. Okay, serine, glycine, histidine are donors of one carbon. Okay, folate deficiency mainly affects DNA synthesis. Okay, because tetrahydrofolic acid is a carrier of one carbon compound, and it is required for formation of C2 of purine, C8 of purine, and conversion of deoxy UMP to deoxy. Histidine metabolism produces which one carbon donor intermediate? Formimino glutamic acid. So histidine load test is given, and if there is folic acid deficiency, so increase excretion of igloo. One carbon metabolism is important for the synthesis of all of the above: purines, thymidylate, and okay. So just check out your MCQ score. If the MCQ score is more than 50%, it is highly appreciable. Less than 50%, so please do rewatch the video and solve all the MCQs. For to get the comprehensive picture on protein metabolism, watch the complete playlist. So this brings us to the end of video series on protein metabolism. So next video series will be on fatty acid metabolism. Thank you for watching. So if you have liked all these videos, please do like, share and subscribe. Thank you. Happy learning.